Well, good day, and everyone, welcome to tetrahydrocannabivarin, THCV, Properties and Promise. Thank you for joining us. First, a disclaimer, almost nothing that we're going to discuss today is FDA approved. The interventions presented in this program are for education and cannot be used for commercial purposes without express written permission of Breeders' Best. If such therapies are applied, outcomes will vary. So this is the story of THCV. So the substance was first isolated in 1970. So this is about 6 years after THC and some of the others. And one of the discoverers was a then very young researcher named Roger Pertwee. And this was his first published journal article in Nature, no less. How often does that happen? Well, never these days. But they isolated THCV from this tincture of cannabis that was part of the British formulary until 1961, actually. So at the time, it didn't have a name, but to quote from the article, there are at least 6 pharmacologically effective components of cannabis. I include that because now the number is far, far higher. So what they described was an N-propyl analog of THC, meaning that it had 3 carbon side chain instead of the 5 that is typical of the pentyl cannabinoids such as THC. Later, it came to be called tetrahydrocannabivarin, or as we'll say, THCV for short. The tested substance was 4 to 8 times less active in producing catalepsy, lack of movement in mice, as compared to THC at 5 milligrams per kilo. Subsequently, this was tested in humans by Leo Hollister, did his work at Veterans Administration hospitals and was the Dean of American Clinical Studies. I'm proud to say that his last article was one we published in the Journal of Cannabis Therapeutics in 2001. But in 1974, he gave 6 humans 7 milligrams of THCV intravenously. 1 reported no effects, 5 described moderate or mild THC-like effects. Based on this, and we don't typically give cannabinoids intravenously, he estimated that THC was about 25% of the potency of THC. So here's the biosynthetic pathway. What we normally see is a combination of olivotolic acid and geranyl pyrophosphate, leading to cannabigerolic acid, which upon decarboxylation would be CBG. And then depending on which enzymes are most active, this can go on to THC in the form of THCA in the fresh plant, CBD, or in a recessive gene, cannabichromene. However, geranyl pyrophosphate can also combine with this precursor, divarinic acid, which again looks like olivotolic acid, but has only a 3 carbon side chain. What results from this marriage are the propyl cannabinoids, the 3 carbon side chains, starting with cannabigerolic acid and with THCA synthase, the same enzyme that produces THC, it can act to produce THCV acid. Again, with decarboxylation, this will be THCV. Now, some studies that were done, inhalation studies circa 2004 or so, these were done under legal conditions. This is the report of one person. 2.5 milligrams of inhaled vapor, and this was a blinded study. Vapor was said to be harsh, with a creepy, meaning slow onset, but with mood elevation, some psychoactivity, a feeling of increased sensory acuity, sharper vision, hearing. When THC preparation was taken later, this seemed to cause a blocking of the effect. Because this was blinded, it was asked what the guess was. It's the substance. The first guess was THC, but later revised. Again, a blinded situation with 10 milligrams of inhaled vapor, was again reported to be harsh, moderately psychoactive with mood elevation. Again, increased sensory acuity, a pleasant feeling, and again, some blocking of THC effect after subsequent administration. In a third application, not blinded, THCV 40 milligrams plus THC 6 milligrams was inhaled with a vaporizer. Again, acrid taste, feeling of initial buzz, never uncomfortable or hard to handle. There was a decreased hunger, an anorexic effect lasting some 5 hours, but with no nausea. The person reported that they were not as hungry as expected in a prolonged fasting state, having missed breakfast. And by lunchtime, the person reported ability to eat, but with no urge, which would be fairly atypical. Scientific study was really only picked up in earnest about 2005. And this is again from Dr. Pertwee's group, who picked this up after essentially a 35-year gap. So they looked at THCV in terms of receptor binding and found that it displaced this agent. CP is a high potency full agonist at the CB1 receptor. So THC was able to displace this with this level. And it also worked on humanized CB2 cells, Chinese hamster ovary cells, with a relatively low concentration necessary. THCV inhibited THC evoked contractions of the vas deferens, implying that it had an antagonistic effect. Now, this next one I will report, but say that I'm afraid it was an error. They thought at the time that THCV was a CB2 antagonist as well, but as we'll see subsequently, this was later overruled. So they looked again at THCV and similarly saw pure substance could displace CP 55940. It also attenuated, reduced THC-induced tail flick, ringing mobility, and hypothermia responses in vivo in mice. They again examined the pharmacology in 2008, and the overall feeling was that THCV is an antagonist of THC and other agonists in CB1 expressing tissues, but also a potent CB2 partial agonist, so weakly stimulating the CB2 receptor, not an antagonist. So this is a very interesting combination that we don't see in other substances. And the first reported natural substance that was an antagonist at CB1, but stimulated CB2. And there are many possible uses for such an agent medically, as we'll see. Riddell et al. Looked at THCV in mice, and they were able to see a dose-dependent weight loss that was highly statistically significant. Food intake was reduced at all doses. However, when THC was present, even when it was outweighed 5-to-1 by THCV, there was a loss of this effect. It was suggested that THC, particularly if pure or relatively pure, could be a possible treatment for obesity. A subsequent study out of Italy showed that THCV inhibited cyclic AMP production by humanized CB2 CHO cells, again, Chinese hamster ovary cells. Diminished a couple of tests related to induction of neuropathic pain. Carrageenan is Irish moss, and when this is injected, it produces swelling and hyperalgesia, and increase in pain responses. So THC diminished both the edema, the swelling, and hyperalgesia at this 0.3 to 1 milligram per kilogram dose given intraperitoneally. THCV also decreased pain behavior in the phase 2 of the formalin test. Formalin, again, produces neuropathic pain. At a higher dose, 5 milligrams per kilogram, it decreased the pain in the initial and late responses. Out of England in 2010, THCV was tested for possible anticonvulsant effects. They used Purkinje cells and it showed a reduction in firing at the CB1 receptor. They tested THCV also in the piriform cortex that's responsible for integration of odor into memory. THCV produced anti-epileptiform effects. It reduced what are called burst complex incidence and paroxysmal depolarizing shift amplitude. And these were shown to occur at what would be possibly attainable doses, concentrations. This concentration of 20 to 50 micromolar is quite high, but many anticonvulsants require high dosage levels, and that's typically true of CBD, so this should not be really a surprise. In pentylene tetrazole-induced seizures in rats, at a dose of 0.25 milligrams per kilogram, which is relatively low, reduced seizure incidence, and 33% of the animals were seizure-free, which was statistically significant as well. In a subsequent study, they used THCV 2 milligrams per kilogram in rats that were treated with lipopolysaccharide or 6-hydroxydopamine. 6-hydroxydopamine is an agent that is toxic to cells in the substantia nigra and will induce a Parkinsonian-like state. So THC enhanced ambulation, the walking of the animals, and their speed, their mean velocity over controls. When given chronically, THCV decreased the loss of the dopaminergic neurons in the nigrostriatal region, is the basal ganglia. It also attenuated microglial activation. So these are in the inflammatory cells. That was highly statistically significant. Both effects were actually enhanced when there was an extract used. THC showed evidence to reduce Parkinsonian symptoms and provide neuroprotection. So that was in 2011, and we'll see that there have been subsequent studies. In this study, they used the synthetic delta-8 THCV, which we assume would be analogous to delta-9. It's possible that this is a little more stable and could be made in the lab. So in mice, at 3 to 10 milligrams per kilogram intraperitoneally, there was attenuation of elevation of serum transaminases. So these are liver function enzymes. And this is done in relation to whether there would be improvement in damage that was produced by a lack of blood flow to the liver. There was also a decrease in tissue oxidative stress that was noted with these carbonyl adducts and 4-hydroxy-2-nonanol. There were also diminished acute and chronic hepatic inflammation markers including tumor necrosis factor alpha, CCL3, etc. Also decreased neutrophil infiltration. And there was also a decrease in necrosis, cell death, and etc. With this intervention. Mechanisms of action seem to be mediated by the CB2 receptor, since they were abolished by a CB2 blocker, SR144528. Once again, Professor Pertwee studied and reported on THCV in 2014. And the feeling at that time, which remains the case today, is that THCV is a partial agonist of CB2 with mixed agonist-antagonist properties, stimulating when receptors are abundant, but antagonizing when less expressed. So that's very complicated and may explain some of the prior confusion. At doses at which THC activates CB2, it also acts as a CB1 receptor neutral antagonist. So this is in contrast to agents like rimonabant, which are inverse agonists and actually knock down the overall tone of the endocannabinoid system. At high doses in some experiments, THCV showed evidence of stimulating CB1. And this would go back to some of the early experiments with high doses where catalepsy, immobility, and analgesia were produced. So again, this requires high doses. It was also noted that THCV was active at TRPV1 to 4 receptors and GPR55, all at 1 to 10 micromolar or less. So these would be possibly attainable in therapeutic applications. John McPartland et al. Did a very useful review in 2015. As you may know, there was quite a bit of controversy in relation to weight loss, anti-obesity drugs like rimonabant that were initially approved in Europe and then taken off of the market because as inverse agonists, they actually knock down endocannabinoid tone and produce not just weight loss, but also depression, anxiety, some suicidality. There were also a couple of cases of multiple sclerosis that were generated. So in comparing THCV to those other drugs, THCV failed to produce nausea in rats like rimonabant would. It also reduced epilepsy responses, as we noted in the Hill study, unlike rimonabant, which actually lowered seizure threshold, made seizures more likely. Little effect was seen in animal models of mood. The dose of THCV that produced anxiety in the elevated plus maze is 30 times higher than that of rimonabant. So there was an obvious increase in safety margin, and it's just not expected that THCV would have the same adverse event profile of the inverse agonist drugs. In another study in human subjects, Tudge et al. Gave 10 milligrams THCV or placebo in separate visits, double blind. They did functional MRI imaging, and responses were monitored to rewards, which included the sight and flavor of chocolate, or aversive stimuli, which in this instance was moldy strawberries. THCV increased the bold responses to chocolate in the midbrain, anterior cingulate, caudate, and putamen. It also increased aversion responses in the amygdala, insula, midorbital frontal cortex, caudate, and putamen. All this suggested that therapeutic application of THCV in obesity would be possible with a lower risk of depression compared to the CB1 inverse agonist. Another study done in 2015, they gave single doses of 10 milligrams of THCV orally or 10 milligrams of placebo in 20 subjects. There were no particular effects seen on mood, energy, or affect. Then there's this paragraph about resting state functional connectivity, etc. It decreased this in the default mode network and increased connectivity and cognitive control and dorsal visual stream network. All this was taken to suggest efficacy in obesity treatment. Jadun et al. Had various treatment arms with CBD or THCV, 5 milligrams twice a day with or without THC, again in humans. THCV decreased the fasting blood sugar. That was statistically significant. It also improved pancreatic beta cell function, the cells that make insulin. Also, significant effect on apolipoprotein A without affecting HDL levels. Interestingly, combination with CBD and THCV didn't produce significant differences. So there was some feeling that although CBD reduces hunger, that the combination wasn't necessarily that helpful. In a subsequent study, humans were given 10 milligrams of THCV versus placebo daily for 5 days with 1 milligram of THCV intravenously on the last day. Only 6 of 10 guessed the assignment, suggesting that there wasn't enough psychoactive effect of the oral THCV to, for people to tell it apart from placebo. There was an increase in intrusions on testing, but also an increase in reverse digit span. So one psychometric measure was reduced, but another one was actually improved. There was an increase in pleasure scores. It was well tolerated. Interestingly, on the last day where THC was given, the effect of it was weakened when it was given with THCV in 9 out of 10. Again, noting some kind of blocking effect on the peak high, at least, of THCV. The impairments, delayed recall from THCV were limited by THCV, and it also knocked down the tachycardia, the rapid heart rate. Very interestingly, in 2016, Etienne Demeyer, who was the chief breeder at GW Pharmaceuticals, reported on how to make more THCV. He noted that wild-type cannabis may contain anywhere from 0 to 2% THCV. They used some chemovars that, to start, had 14 to 69% of their total cannabinoids as THCV and crossbred them. And this was the scheme to do it. So breeders might want to get this paper and study it. But through this inbreeding from multi-cross hybrid combinations, they were able to attain a situation where 96% of the total cannabinoid content was as THCV. Using the same scheme, they were also able to produce very high amounts of CBDV, CBGV, and CBCV. And it was predicted that field yields of THCV could be 60 grams per meter squared, which seems to be very generous. And that was lower for some of the other agents, particularly CBCV, which again is only produced via a recessive gene. A very interesting study out of the Gardner lab at the National Institutes of Health. They again used delta-8 THCV, and it reduced intravenous nicotine self-administration in rats. Also, Q-induced, nicotine-induced also reduced nicotine-induced relapse to nicotine seeking in the animals. In mice, it also attenuated nicotine-induced condition, place preference, and withdrawal. So their conclusion was a new and potent anti-nicotine pharmacotherapy for humans may evolve based on medicinal cannabis. Keeping in mind that nicotine is one of the most addictive substances known to man, the same would likely be true for other treating other drugs of addiction. In the 2020 study, again, it was Parkinson's disease was examined in an animal model, what's called PIT-X3AK mutant mice that have a dopamine deficiency. They were treated with THCV 2 milligrams per kilogram intraperitoneally over the course of 2 weeks. These were animals in which dyskinesia, abnormal movements, were elicited by L-DOPA treatment on measured abnormality, abnormal involuntary movements or AIMS. THCV significantly delayed these involuntary movements, reduced their duration and intensity with significant decreases in previous elevated levels of phospho protein and histone PACH3. THCV attenuated the intensity of the dyskinesias within 3 days, even when administered 2 weeks after the L-DOPA treatment. There have been no clinical trials of this in Parkinson's disease, but certainly this provides a foundation for that effort. Now, let's compare THCV to CBD. So THCV and CBD both attenuate THC-induced sequelae, side effects. THCV is a neutral antagonist and CBD is a negative allosteric modulator. What that means is CBD binds to another site on the CB1 receptor and alters the binding of THC. As we've seen, both affect TRP channels, although sometimes different ones. For example, CBD seems to be much more powerful at TRPV1, where capsaicin, the ingredient of chili peppers, works. THCV is seemingly much more potent than CBD. What that means is only that fewer milligrams of THCV are needed to be effective for a given role therapeutically. Both seem to have roles in treatment of pain, inflammation, and epilepsy. Both seemingly have anti-addictive effects. And for both, when growing them, it remains difficult to eliminate THC completely from THCV and CBD chemovars, but there's always more trying. Certainly, there'll be people that are interested in genetic modification, but I feel that's not necessary because selective breeding with conventional means can get you very close to or all the way there. Comparing THCV and THC, THCV seemingly would be a nice adjunct to any THC chemovar by complementing its therapeutic properties and attenuating sequelae. This is another way of saying increasing the therapeutic index, the ability to take more THC with fewer side effects. What about economic prospects? The unique properties of THCV seem to offer great promise in treatment of type 2 diabetes, obesity, the metabolic syndrome, addiction, and Parkinson's disease. Certainly, all of these are areas that require more effective and safer products. I feel that the safety and versatility of THCV make it a very attractive addition to various cannabis-based preparations. And I believe that's it. So I will turn things back over to Nisha and we will address your questions. Thank you.